Hi everybody, this is Dr. Karens, and in this video I'm going to be going over the discussion and conclusion section of your unknown project lab report. So this might be the hardest section to write, particularly if you had a really easy time with your experiments and identifying your unknown. Um, it's kind of hard to discuss a lot when things worked perfectly. But let's talk about what kinds of things you should be addressing in this section. So first of all, this is where you're going to describe how you determined what tests you were going to conduct. Um, I thought about what section to put this in, and I think I want to put this here. So it's up to you how you decide what biochemical tests you think you're going to need to identify your bacteria, but this is where you're going to discuss that. It might only take a few sentences, but that's the first thing I want you to address in here. Then I want you to explain how the test results led to your identification. And this means walking the reader through your dichotomous key. Now, I don't want you to repeat anything about procedures or any lengthy descriptions of results. I want this to be succinct. For example, the first test that everyone's going to do is their gram stain. So that's where you would start. Say the gram stain revealed that um, the unknown was gram negative. So that led to this first test on the key, which is I don't know, I'm just guessing. Let's say phenol red lactose. That test turned out to be positive. So that led to this test on the key, which turned out to be negative, which leads to this test, which turned out to be positive, so on and so forth until you reach the identification on your key. So that's what I mean by succinct, but you're still going through the key step by step. Other things that you can discuss here and should discuss if it's applicable. Any variables in your results or reasons why your results may not match what is in the book. And so this is where you can discuss any problems you had during the experimental process and how you solve them. You want to compare your results to both the standard results that are in the book and to the abnormal class data, and then discuss any discrepancies there. So mention any discrepancies, and then you need to investigate why there were discrepancies in those results. This is going to mean that you are going to have to do some research. You need to do a valid search, meaning you need to spend some time looking, because it's possible that there are documented reasons for why, E. coli turned out to be positive for a test when it should have been negative, for example. There might be documented reasons somewhere. There might not be, but I want you to put in some time and do some digging here and talk about that. If you found yourself trying to decide between two organisms, discuss how you finally made your decision. Again, addressing inconsistencies between standard results and class results, even. Um, if you did two tests that both test for the same thing, did you get the same answer? So one example is PR lactose tests for lactose fermentation, as does EMB. Now you may or may not have decided that you wanted to try some other tests beyond the biochemical tests that were in the book. And that's fine. You don't have to do that. I'm not asking you to do that. But some students are who are maybe a little more astute are thinking about, wait, there are other tests that we've done earlier in the year. Maybe I can do some of those. I can't guarantee that you can because I don't know if we'll have the reagents ready. But for example, again, EMB and PR lactose, they both test for the same thing. If you did both of those tests and you got different results, you definitely need to talk about that. Let's see. If you decided to do all of your biochemical tests, all of the tests that are in the book, but you only used some of them in your key. When you walk through your key, you're obviously not going to address all the tests then. You're only going to address the tests that are in your key. But if you did all the tests, this is where you can talk about the tests that were not in your key. How did those results compare to the standard results? How did those results compare to the class data? Talk about those things. Let's see. If you came to a conclusion about your microbe's identity despite abnormal results, explain the reason for your conclusion. You need to sell this. You need to sell this to the reader that even if you got results that were unexpected and don't match what you expected to see, how do you know that you have E. coli? How do you know that you have SRAs? You need to sell this. You need to be confident and you need to give me the reasons why. Please, please, please include references here if you need to. Most people should be trying to do some digging into finding out, especially if you have discrepancies, do some digging and see if you can find out why. Restate the full name of your organism here. Here's what I don't want in this section. Do not, I mentioned this earlier, don't give me descriptions of your methods and lengthy descriptions of your results. Like, I don't want to hear again that your PR lactose test was yellow, which means it's positive. You already told me it was yellow in your results section. Just restate the result, positive or negative, as you're going through your key. Don't assume that whoever is reading your paper has any access to the data that you're referring to, except what you provide in your paper. So, some things. So, like if you are referring to class data, make sure you state what that class data was for that specific test. I need to know what that is because don't assume that I have access to that information. If you're going to refer to your tests, which you will be referring to tests in this section, the test that you did, make sure the results of those tests are in your results section. That, again, I've had students talk about tests in this section that are not in their results section. And I had no idea that they had even done those tests. So, again, any test that you did should be in that results section so that when you want to talk about them, you can talk about them here. And then finally, going back to how do you explain inconsistencies, don't just fall back on the old, it must have been contamination or user error. That's an easy way out. I want you to take a legitimate effort to look up why there could be some inconsistencies in your results. It's possible you might not find any documentation about that, but you could state that in the paper. You could say, research into why this turned out to be negative was inconclusive or not, results couldn't be found or information couldn't be found about why. So it's possible. That's when you can say possibly maybe there was contamination, but I would be a little more specific with that. Like, what do you mean? Like, what could have contaminated it to give you the wrong answer, to give you the wrong result? I want a more intelligent and thought out answer than just contamination. Okay. All right. So the last bit of the instructions document talks about references and some other things, and I will do that in the last video.